right, we're gonna continue our program. Our next speaker is Blair Leavitt, who comes to us from University of British Columbia with a very exciting topic of the presentation of using lipid nanoparticles in CRISPR gene therapy for neurologic disease. And I suspect that neurologic disease that you know one.
Got it. Couldn't find the clicker. So thank you very much. Thank you, Emily and the Bao Foundation, for this opportunity to talk today. This is my first GNO1 conference, so be gentle. By way of disclosure, I'm going to be presenting a novel delivery system that was developed in my lab but is being commercialized by a company that I'm still involved with, Incisive Genetics. I'm going to be presenting some non-confidential data, but I'd like that to be a disclosure that's noted. Like many of the scientists in the room, I got into GNO1 research because of interactions with a local family. I think they came to me not because of what I do in the lab, but because of what I do in the clinic, where we've done some of the first gene therapy in another brain disorder, Huntington's disease. We were specifically the first site to administer an antisense oligonucleotide therapy, and they came to me hoping that the antisense therapy would be appropriate for their child. I told them I didn't think so. I thought they needed curative gene therapy, thinking that maybe I was getting out of things. But unfortunately, they persisted and asked whether that was something that was actually possible. Certainly it was science fiction not that long ago, but as I think everyone in the room probably knows now, genome editing is really coming to the fore. We have an approved therapy based on CRISPR-based genetic editing. And Pardis is going to tell you a little bit about how we're applying this to GNO1, but I'd like to introduce a little bit into the approach of how we're delivering CRISPR-based genome editing to the brain. So this is our delivery system. It's a non-viral delivery system. We're using lipid nanoparticles, which you may not all know about, but you've probably all had. This is what was used for the mRNA vaccines, the Pfizer and the Moderna COVID vaccines. These are a non-viral way of delivering various forms of genetic material. They work incredibly well with nucleic acids like mRNAs and other. What they don't do so well, or at least the traditional way of manufacturing them doesn't do so well, is deliver more complex therapies such as proteins or protein complexes. And what we identified in the lab was a method of encapsulating very complex therapeutic modalities into very standard lipid nanoparticle compositions. So we have a unique process that allows us to encapsulate what the RNP, the ribonucleoprotein, and I'll tell you a little bit more about what that is. We've enhanced the way that they can disrupt and correct genes. We can do a form of gene correction called homology-directed repair, which when we started this, I was told doesn't happen in neurons or in postmitotic cells, but it clearly does at high levels. And we're using LNP compositions that are very similar and safe in human. And importantly, they can be repeat dosed. So I'm going to tell you about an approach that allows for titratable genome editing. So CRISPR-based editing, this is a very powerful tool that we can change the DNA sequence both in vivo and ex vivo. At its basis, we need two things. We need an effector protein. Cas9 is an example. It's a scissor-like protein that can cut DNA. It can be modified with all sorts of approaches to manipulate DNA, to methylate, to do other things. And it needs a guide RNA, which is an RNA that targets to a specific sequence in the DNA. And it can target any sequence in the genome, and different effectors can target different regions with different efficiencies. But the effector, what actually works, is the ribonucleoprotein complex. You need the protein bound to the guide that can then target the DNA and do whatever it is your effector wants to do. Cleave it, methylate it, whatever. So it's really this RNP that needs to get into the cell at high enough levels to be effective. And it's been known for a long time. Dr. Dudna, Nobel laureate, has said this over and over in many different talks. Delivery is the key. And there are a number of approaches currently being used. We can use nucleic acid. We can deliver the individual nucleic acids with lipid nanoparticles, much like the vaccines. But then you're delivering the individual components. They have to be made in the cell. They have to be made at high enough levels to form the complex. Then the complex can act on the DNA to make a permanent change. Or you can use virus, but virus has other limitations in what you can deliver. It again is delivering the nucleic acid, and you might have uncontrolled production of these enzymes unbound to their guides, which can potentially be harmful. We've known in the gene editing field for a long time that RNP is the gold standard. We've done this for many years. When you take cells out of the body, you can deliver RNP through a variety of techniques like pronuclear injection to make mice, which is what we were using it for. But you can also use electroporation or other tools. And that's actually the way that the first FDA-approved CRISPR therapy is done. Stem cells are taken out of the body, they're manipulated, and the RNP is delivered to the cells. They're selected and delivered back through a bone marrow transplant. And that's the therapy called Casgevy, approved for thalassemia and sickle cell. So what we do is we've identified a way of encapsulating the fully complexed RNP. So this is the enzyme, and most of the data I'm going to show today is Cas9, but it can be any enzyme, any endonuclease, bound to its guide. So it's pre-bound. There's no free enzyme delivered. And as well, we can have multiple guides in a single formulation, and we can also include DNA templates, which allows homology-directed repair, as I said. So these LNPs, if you can deliver them to cells, are very effective at getting taken up to cells. So they basically co-opt the cell's cholesterol delivery system. We've all had our HDL and LDL measured in our blood. The LDL particles are the cholesterol delivery system to cells. There's a receptor, an LDL receptor on the cell. They recognize these LNPs as cholesterol delivery mechanisms and endocytose them into the cell, where they're then released. The RNP is released and can enter the nucleus and target the DNA. Simplified, but I think that's the basics. I'm not going to go through the key advantages in great detail other than to say it's a non-viral approach. It acts immediately and then is rapidly cleared. We have evidence in ex vivo systems that DNA editing can occur within hours. Once it occurs, the DNA changes are permanent and persist even through multiple cell divisions. Unlike AAV, the cargo is protected from the immune system in the circulation and in the tissues, so you don't develop antibodies, and repeat dosing has now been shown not just in vivo in animals, but the first demonstration of delivery of LNP-mediated CRISPR has been shown in humans now. So, and also, unlike more complex approaches such as viruses, it's easy to manufacture in high doses. So I'm just going to show you some of the key studies, and then I'm going to pass it over to Pardis, who's going to tell you what you're really interested about and how we might use this for GNAO1. So I mentioned that this is highly efficient and much more efficient than delivering mRNA. This is a very simple system, a dividing cell line, HEC293 cells, and this is sort of our standard, what we use as our quality control for our LNPs, targeting a gene, Huntington gene, that's my favorite gene. We get about 50% editing very reliably with a very small amount of Cas9 protein. We cannot get the same amount of editing with mRNA, even when we go up to 10-fold increases. And this is in a dividing cell type. When we go to neurons, we can't get any editing at all when we deliver the mRNA. And I'm going to show you very effective editing when you deliver the RNP. So the key is this allows very small amounts of very effective genome editing to be delivered in a safe and well-tolerated way. This is just some examples showing that neurons can be effectively targeted. This is gene disruption, my favorite gene again, Huntington, targeting for gene disruption with LNP delivery of RNP targeting the gene. You can see here the Cas9 protein stained red within the neuron, translocated into the nucleus, the blue cell in the center. And we get very efficient genome editing in neurons. That's ex vivo primary neurons cultured from mouse cortex. We can also do it in human cells, human iPSC-derived neurons, also highly efficient at genome editing. Again, the Huntington gene knockdown. And we can do this in vivo. So we've done this now in vivo, direct intrastriatal injections of LNP, RNP into the striatum and effectively targeting the Huntington gene. In this case, we did one site with 2 microliters versus 2 sites with 1 microliter each, same total volume, same effect. We were just looking at distribution of different delivery approaches. But as I said, we're not trying to disrupt genes when we think about GNA01, at least not in the model that we're looking at. We want to correct the gene. So we're using an approach called homology-directed repair. We've done other approaches, such as base editing, that also work with this delivery system. But we can use good old SP-Cas9, deliver it as a preformed RNP with the a DNA template, and get highly efficient homology-directed repair, even in post-mitotic cells like neurons. So to show this, we're using a very beautiful system developed by a colleague. This is mutations in a gene called PAX6 that cause an eye phenotype, aniridia. She developed this model in mice where the mutation is linked to a flag tag. When the mutation is present, there's no production of the protein, so if you stain with the flag tag, you don't get any staining, but you do still produce the normal amount of PAX6. So these are relatively normal mice. But when you correct the mutation and you get production of the protein from the mutant allele, you get now flag tag protein that can be identified with antibodies. And it's a little bit dark here, but in the control, there's absolutely no flag tag. You can see the PAX6 staining from the normal allele. And we get highly efficient, and this is in primary neurons again from the mice, correction. These are every single red cell here is expressing PAX6. It's expressing the protein from a mutant allele that's been corrected using RNP, LNP delivery. We can quantify this, and it's dose dependent. As we go to higher doses, we get increased levels of HDR. And importantly, we can do repeat dosing. So we can administer and then administer again, and we see an additive effect and increased levels of homology-directed repair. Again, opening up the opportunity of titratable genome editing in vivo. We've already been introduced to the TD tomato system. TD tomato is a red fluorescent protein. There are mice that have this behind a bunch of stop codons. Basically, if we disrupt the stop codons, we can make the cells turn red. So this is just a nice biomarker for genome editing in vivo, and we use it to look at biodistribution and delivery of the RNP, LNP to various cells throughout the brain. We've been able to show beautiful delivery to various cells through direct delivery into the cornea. We get nice distribution of red cells. Intraparenchymal delivery in the brain gives a nice distribution. This is the striatum. Again, I showed you the quantitative effect on targeting a gene. This is the distribution of genome editing. Direct intrafemoral injection allows us to target bone marrow stem cells in vivo. And like all lipid nanoparticles, if you deliver it systemically, most of it goes to the liver, and we get very efficient delivery to the liver. We have moved now into non-human primate. This is staining for Cas9. We do have genome editing data that is coming in non-human primate, but this is just showing that we can deliver, and we can effectively target a part of the striatum, the putamen, in a non-human primate. So this is a brief introduction to the delivery system that Pardes is going to use in the GNA01 model systems to show homology-directed repair and correction of a specific GNA01 mutation. So this allows effective delivery of genome editing to multiple cell types and tissues, both ex vivo and in vivo. I've shown you some of the Huntington data. We can effectively target that and reduce the level in neurons. Homology-directed repair by HDR is both effective and safe and occurs both in the liver and in the brain in post-mitotic cells at high levels, which is actually contrary to much of what you might read in textbooks. We've shown effective delivery of various endonucleases, Cas9, Cas12, base editors, etc. There's low toxicity, immunogenicity. We have done in vivo repeat dosing, and again, it's safe for in vivo delivery. So, without anything further, I'm going to thank the members of my lab, our collaborators, former lab members, and part of my team at Incisive that actually were two members of my lab that now are the key principals at Incisive. And I'm going to pass it over to Pardes, who's going to tell you about GNA01 stuff, which is what you really care about.
Thank you. Yeah, perhaps that's what we should do. We should just hear from Pardes next and then ask you the questions, both of you.
Well, yeah, I don't want to take any. She'll answer her questions because she's
Okay.
smarter than I am, but I'll answer any questions about the delivery.
Fantastic. Then let's ask the questions about delivery only.
this is going to sound like a naive question, but based on how they work, do you think you could titrate the levels of uptake by giving a person statins?
It's an excellent thought. Yeah, we've actually looked into that because. Oh, I'm sorry. I'm sorry. Absolutely, it's a really interesting thought, and we have looked at the potential role of LXR antagonists. I can't remember. Statins is a good example of increasing low-density lipoprotein receptor as a means of increasing delivery. Absolutely, it's a great thought, and it's one that we're really interested in.
I have a very basic question. So there are about 100 billion cells in our brain. So how many of those cells do you think you can repair? And how many of those cells in the patient brains do you think need to be repaired to show the therapeutic effect? And can you match these two numbers?
It's an excellent question, and one that will be answered probably empirically. What actually got me most excited about this when we first came up with the idea was the fact that it's titratable, that we can repeat dose. All of the current therapies, at least at the time, were one shot, where you've got to get your maximal delivery in a single injection. We're already doing injections in my field and Huntington's disease field, direct intrastriatal delivery of various gene editing or gene therapy approaches. We don't know, and we won't know until we generate more and better empiric data. I think the fact that, again, what gives me hope is that if we can get 10 or 15% of cells within, say, the striatum or caudate putamen in human effectively edited with a single delivery, and we can show, as we have in model system, additive delivery. If we need to get to 50% or 60% or 70% of the cells within a target region, hopefully by repeat dosing we can get there. It remains to be shown.
So I was super excited that you could show that you get HDR in postmitotic neurons. Could you tell me how that works then? Because normally it's you need cell division, right, for homology interaction.
Yeah, and we've compared a number of approaches in postmitotic. So these are the HDR that we're showing is in primary cortical neurons derived from mouse brain. We're administering it at a time when the cells are no longer in division, both with base editor and with Cas9 HDR. We're seeing very high levels. I think that the data suggesting that you need cells to be in division is simply wrong. If you're delivering the preformed complex, probably at least in proximity, if you're not in some way in a complex with the DNA template, the efficiency is high enough that you're able to see it in these cells, whereas with previous systems it wasn't. So there have been other situations in which HDR has been shown in neurons, where it's expressed at very high level, for example, from virus. And if you get enough of the Cas9, enough of the guide, enough of the template into even a postmitotic cell as individual components, you then start to see HDR. So I think it's a limitation probably of the systems that were looked at in the past. When you deliver it as a preformed complex and it's immediately translocated to the nucleus and can act, I think it's just efficient at a level that we hadn't previously seen. That's my take. We have done in vivo delivery, and we can show it in cells within postmitotic cells in the liver as well. So it happens in vivo, it happens in primary cells, and I just think that the idea that postmitotic cells don't undergo HDR is wrong.
Okay, thanks.
If I may follow up on this, because like our lab's been doing for the last couple of years a lot of the HDR with the LNP particle, not LNP, but like just ribonuclear protein delivery by electroporation in the brain. You have to go like really in vivo for this to work. You got to go in embryonic stages, and only then you see the different neurons in which the editing happens. We see only if the neuron was mitotic. You see efficient editing. So the moment it's a post-mitotic population, it's just abysmal. So there's got to be like some sort of a secret sauce. Maybe that's your lipid coating that makes like, but there's a nuclear membrane.
I think it has to do with the expression of the nucleic acid. So we've spent a ton of time, and again, most of the LNP work is done delivering nucleic acids. Highly efficient delivery of Cas9 mRNA into post-mitotic neurons, we can show it doesn't make protein. I don't know why. It very inefficiently.
Yeah,
So delivering the preformed protein seems to bypass that step, and I don't know. I don't understand it.
I'm talking about RNP particle delivery by
Oh,
electroporation.
RNP.
Yeah. So like premix everything. It's exactly this except for the lipids.
Well,
Yeah. So maybe lipids, that's what I'm guessing. Maybe
Maybe.
lipids make some sort of a difference.
So question, when it comes to your, when you design your guide for CRISPR, is the vision to have like, so, because we have these hotspot mutations, right? Or there's like a region where a bunch of mutations put together. So how do you envision this to benefit multiple patients, not, I hope it's not patient specific.
so
And then, sorry, one more.
Pardis is going to talk about guide screening and
Okay.
our strategy. I would say that, I've presented some work from a company that's commercial. The work being done on GNA1 is academic, and how small, this is going to be personalized medicine. How we translate personalized medicine, broadly speaking, is still an unanswered question.
Gotcha.
We're
And-
doing this as an academic exercise, again, brought into this by the families and the need to develop it, but it's not a commercially viable approach yet. So I don't have an answer. The field doesn't have an answer. How are we going to really do this when we need individualized therapies? It may be that regulatory agencies need to recognize that if you hold all the components constant and are simply changing the sequence of a guide RNA, it's essentially the same components, and perhaps if we get to a point where we can approve an approach and change the guide to target different regions without going through all of the steps needed for approval of a drug, we can get to a point where we can do that. That's what would be required. In order to do therapy for every individual with GNA1, we're going to have approaches that target individual regions in the genome. I don't see a way around it.
Let's talk about it later. What we're doing in our case is that we're just doing indiscriminate silencing of GNA01 and then overexpressing it again. This way we don't have to design it for specific point mutation. But irrespective of that, toxicity, that was the last question I wanted to ask, because you said you could do repeated delivery, high doses, low doses. Is there any tox observed in non-human primates or mice?
So far we haven't seen any. We've looked very closely for any evidence of inflammation, neuroinflammation, or systemic inflammation, but not to date. I mean, examples of similar compositions of lipid nanoparticles delivering mRNA have been repeatedly delivered. Onpattro is a systemic delivery system for an siRNA, and it's been delivered very safely to thousands of times to patients. So,
Thank you very much.